My name is Mercedes Herrera from the University of California, Irvine. Today I'm presenting correlating highly differentiated cytotoxic T cells with NT5c1A antibody status and disease severity in participants with inclusion body myositis in the INSPIRE IBM trial. IBM is a slowly progressive muscle condition associated with a blood autoantibody known as NT5c1A and highly differentiated cytotoxic T cells. Many CD8 T cells express a surface marker called KLRG1, and these cells have been hypothesized to have a pathogenic role in IBM. These specialized inflammatory cells are thought to be responsible for disease progression and unresponsiveness to conventional immunotherapies. Our preliminary data correlated a higher frequency of CD8 T cells in IBM patients who had more severe motor weakness. There's a robust need to further investigate how these cytotoxic T lymphocyte populations can influence disease severity in a larger cohort, because understanding these potential relationships can shape the trajectory for pivotal, more targeted therapies to treat IBM patients. We analyzed data collected from the INSPIRE IBM study, which is a prospective NIH-funded observational study that is ongoing involving 120 participants with IBM. At the baseline cross-sectional visit, we collected serology for NT5c1A antibody status and 8 milliliters of blood for flow cytometry analysis. Functional assessments collected to measure disease severity were the Inclusion Body Myositis Functional Rating Scale, known as the IBM FRS, SIBM Physical Functioning Assessment, known as SIFA, manual muscle testing, also known as MMT, the timed get up and go, commonly referred to as TUG, Sydney Swallow Questionnaire, and EAT-10. Flow cytometry was used on PBMCs to analyze the percentage of immunosenescent lymphocyte staining for CD8, KLRG1, TEMRAs, and Tregs. Below you can see the Treg panel, which are regulatory T cells that help our immune system. We used simple linear regression models to compare the highly differentiated cytotoxic T cells with functional assessments. Our findings show a significant relationship between the frequency of CD8, TEMRA, KLRG1, and more severe functional severity reported on the IBM FRS, SIFA, MMT, and TUG. There was also a strong correlation between the expression of Tregs HLA-DR and more severe functional severity reported on the IBM FRS, SIFA, MMT, and TUG. HLA associations may affect disease behavior, specifically more severe muscle weakness with the HLA-DRB1-0301 genotype. Lastly, there was a slight negative correlation between the frequency of activated Tregs and the frequency of KLRG1 CD8 TEMRAs. This is expected as a higher presence of Tregs is associated with a lower frequency of CD8 T cells. Out of 120 participants, 48% were seronegative and 51% were seropositive to NT5c1A autoantibodies. We ran 19 two-sample t-tests to correlate T-variant lymphocytes with antibody status. We used a Bonferroni correction for p-value significance, resulting in a threshold of 0.0025. Our analysis did not show an association between T-variant lymphocytes and serological status. KLRG1 is increased in IBM patients compared to healthy controls and a potential drug target for IBM. Previous studies have suggested that a consequence of ineffective IBM treatment strategies is their inability to address the shift in IBM blood and muscle to a highly differentiated cytotoxic T cell phenotype. Understanding the relationship between highly differentiated cytotoxic T cells and disease behavior could provide an advantageous approach to IBM treatment. INSPIRE IBM will be investigating these measures over a 2-year period. To conclude, we plan to analyze the frequency of muscle-invading T cells on immunohistochemistry compared to the frequency distribution of cells collected from blood samples through flow cytometry. Lastly, here is a list of my references. If you're interested in learning more about the INSPIRE IBM study, I suggest heading over to the NIH website. And if you're interested in seeing some of the projects we completed last year for this study, you can find those in the Neuromuscular Disorders Journal. Thank you for listening and thank you for your time.